discussed about how DNA replicates. Now, in this lecture, we will be discussing how to make proteins from DNA, or another way to say that is to how to express the genes encoded in the DNA. So, gene expression is the conversion of the information to the gene's product, which is your protein. So, transcription, we have actually two steps in gene expression. We have transcription that converts information in a gene to RNA. And we have translation that converts information from your RNA, well, basically your messenger RNA, to a protein. So, just a brief recap. So, we have your DNA. So, each DNA strand. So, DNA is a double-stranded molecule. And each strand consists of a chain of 4 nucleotides. We have adenine, thymine, guanine, and cytosine. And the sequence at which those 4 bases are arranged is the information. And we call that information the genetic code. Now, all of the cell's RNA and protein products are encoded by the DNA sequences called the genes. Now, your DNA is a very large molecule, and we have specific segments of there that encodes for a particular protein product. And those regions that encode for a protein product are called genes. So you can say that genes are part of the DNA. So the DNA contains the genes, and not the whole DNA is the gene, basically. So basically, it's just a small part. The gene specifically is the part of the DNA that encodes for information, a specific information, or for a protein. So DNA is first transcribed to RNA. RNA, or ribonucleic acid. So we have enzymes synthesizing a complementary strand of the RNA from a DNA template. So the RNA comes from the DNA. So what's the difference between DNA and RNA? So RNA is mostly single-stranded, and it uses a ribose sugar instead of a deoxyribose sugar. So what's the difference between a ribose and a deoxyribose? Well, both of them are ribose. That means it's a 5-carbon sugar, but DNA has the oxygenated second carbon number 2. So basically, the carbon in position number 2 has no hydroxyl group for a DNA. That's called deoxyribose. Whereas for a ribonucleic acid, the RNA, the hydroxyl group on that second position still exists. And it's actually one of the reasons why RNA is less stable than your DNA. Now, RNA, another difference is that RNA uses uracil instead of thymine. So, if you remember the 4 letters A, T, C, and G, instead of the thymine T, RNA uses uracil U. But still, the base pairing is retained. That means adenine. Now, for RNA, adenine is pairs with uracil. So, this is a more detailed comparison. So, the DNA is a double-stranded structure that forms a double helix, whereas your RNA is a single strand. Although it doesn't always base pair on its own. So, it usually folds over. It's like RNA is actually similar to proteins. It has its own specific folds. So, the monomeric building blocks of the DNA are the deoxyribonucleotides, whereas for the RNA, you have your ribonucleotides. And then for the sugar, it contains the deoxyribose, as I said a while ago, and then for the RNA, it's ribose. So, the bases, we have the 4 bases, A, T, C, and G for the DNA. And for the RNA, we have uracil instead of thymine. So, adenine, guanine, cytosine, the same, but uracil instead of thymine. Now, for the base pairing, A pairs to T, G pairs to C. For the RNA, it's A pairs to U instead of T, and then G pairs to T. So, for the function, DNA is mainly involved in storage of genetic information, whereas your RNA involves in protein synthesis and other roles. So, we have transfer RNA. We also have regulatory elements such as microRNA, small nuclear RNA, and something like that. So, RNA has more functions than your DNA. Well, as of the current research. Now, these are the images of the bases of the DNA and the RNA. So, the differences you have, for instance, uracil. This is the base uracil for the RNA, and this is the base thymine that is used in DNA. So, as you can see, they are very similar. The difference that thymine has its methyl group here, uracil has no methyl group here. But generally, they are the same. That's why uracil can also base pair with your adenine. And then you have your ribose sugar. So as I told you, the second carbon, the carbon in the second position contains hydroxyl group in the ribose, but it's absent in the deoxyribose. And then you have your well, basically, this is your adenosine triphosphate for an RNA and deoxyadenosine triphosphate. This is how you form nucleotide. So the nucleotide, when you look at the nucleotide, the difference is on the OH group here. So there's no OH group. This is for the DNA. That's why it's deoxy, without oxygen. Okay, now in RNA, there are three major types, but the types of RNA is not only limited to these three. We have many different, actually current research, they are discovering more and more smaller and smaller RNA molecules that are present, usually in regulatory mechanisms, how silencing genes, mostly on silencing genes and such that. But anyway, these are the main three, and these three are involved in the gene expression. So you have your messenger RNA. Well, by its name, messenger, it is basically the messenger. It's the one that transfers information from the DNA to the ribosome. So basically, it contains the information transcribed from the DNA. And then we have the ribosomal RNA or the rRNA. So it's the main component of your ribosomes. And the ribosome is, if you remember from the parts of the cell, the ribosome is the site of protein synthesis, they call it. But in reality, it's actually the one that synthesizes the protein. So the ribosome synthesizes the protein. It builds the polypeptide chain. So the main component of the ribosome is your ribosomal RNA. Basically, ribosome is made up of rRNA and a few proteins. And then you have your transfer RNA. So tRNA, so it's basically a T-shaped molecule that delivers amino acids to ribosome. So ribosomal RNA and transfer RNA has its roles in translation, whereas messenger RNA has its role in both transcription and translation. Now, in your gene expression, so you have mRNA. So mRNA is translated to protein. So as its role is in the messenger, it copies information from the DNA, and then that information is translated to protein. So you have rRNA and dRNA to help translate the sequences of your base triplets in mRNA into a sequence of amino acids. And the base triplets, we call it codon. So in the translation step, the information carried by your mRNA is decoded into a sequence of amino acids. So this results in a polypeptide chain that folds into protein. So basically, when you talk about translation, it's about translating the three base code, the codon, into your amino acid protein. So if you remember, amino acids, you have 20 amino acids to build a protein molecule. So, and then if you look at your mRNA, we have 4 bases. So you translate the information from the 4 bases, the sequence of 4 bases, to the sequence of 20 different amino acids. So basically, that's why, it's translation. So the DNA sequences, which is the genes that contain all the information needed to make molecules of life. So gene expression is a multi-step process. So we have transcription and translation. That is to simplify matters. But in reality, we have regulation every step of the way. So genetic information from the genes is converted into a structural or functional part of a cell or an organism. So first, let's discuss about transcription. Transcription is the process of creating or synthesizing RNA from the DNA. So in this case, we like your replication, we have a polymerase to do the job. But in this case, instead of a DNA polymerase, we are going to use the RNA polymerase. So the RNA polymerase assembles the RNA by linking the RNA nucleotides into a chain in the order dictated by the base sequence of the gene. So you're basically just copying what the gene says, or rather, creating the RNA as a complement of the DNA. So the new RNA strand is complementary in sequence to the DNA strand from which it is transcribed. So DNA replication and transcription both synthesize new molecules by base pairing. But one of the differences is that uracil is used instead of adenine. So when, for example, in the template, you see a thymine, or rather, because the template is a DNA, so there's a thymine there. So instead of adding, if there's an adenine in the template, instead of adding thymine, because adenine base pair with thymine, the RNA polymerase added uracil. So that's how it goes for transcription. So and then we have your RNA polymerases doing the job instead of the DNA polymerase. So here is a base pairing for the DNA replication versus RNA transcription. So the one at the bottom is our template. So in the DNA, you just add the bases as they pair together. So G is paired with C, T is paired with A, so G pairs with C, T pairs with A, G, so on and so forth. So this is for DNA replication. Now, for RNA transcription, you have, again, the same strand, but you're just going to add the same base pair. You're just going to follow the same base pairing, except that when you go to adenine, when it encounters adenine, instead of adding thymine, you just add uracil. So that's for RNA. So, and then, so same goes, G pairs with C, and then again, adenine is paired with the uracil. And then if you see thymine, you add adenine, of course, because adenine is paired with thymine. So it's just that in the RNA transcription, instead of using thymine whenever you encounter an iodine to pair it with, you're just going to add uracil instead. So that's basically the difference in transcription and replication. So the RNA polymerase and regulatory proteins attach, the first step of transcription, you have RNA polymerase and regulatory proteins attaching to a promoter, which is a specific binding site in the DNA close to the start of a gene. So again, a promoter is another region. So if you have the genes as regions in the DNA that encodes for the information, or the part that we need the information to create a protein, we have another region, upstream. When we say upstream, because it's based on the direction of the read. Now, it's on the upstream side. So you have a promoter sequence. So it's a region that tells the DNA, or rather the RNA polymerases and the regulatory promoters, where to start, or it's basically just like so it's like a landmark to attach there so that you will know. Usually it's actually 10 nucleotides from that promoter site to the actual start of the gene. So it tells the RNA polymerase where to start. So the RNA polymerase moves over the gene in a 5' to 3' direction, similar to how your DNA polymerase goes. So it unwinds the DNA helix. First, it must unwind. Of course, just like in replication, you unwind the DNA helix, and then you move 5' to 3' direction, reading your base sequence. So you are adding bases as you go. It joins free RNA nucleotides in the complementary strand of your mRNA. So here is a view of that. Now, this is your promoter region. The RNA polymerase will bind to the promoter region, and then this is your gene sequence. And then your RNA polymerase will then start transcribing. Now, during transcription, again, the double helix must unwind because you need to access the bases. And another thing is that for transcription, it's just only a single strand. So you are just going to use a single strand, whereas your in replication, both strands are being replicated. In this case, for transcription, only one strand is being transcribed. Okay, so you add your bases as you go along. So there, the RNA polymerase is adding the bases as it moves in the 5' to the 3' direction. So after transcription, you have what you call the post-transcriptional modification. So RNA is modified before it leaves the nucleus as the mature RNA. So in eukaryotic DNA, or rather eukaryotic genes, you have introns and exons. So basically, the genes for eukaryotic cells, it's made up of regions called introns and regions called exons. So introns are nucleotide sequences that are removed from the new RNA strands, whereas your exons are sequences that stay in the RNA. So another way to say that is, for example, if your transcript, so after transcription, you have a raw transcript that must be edited, removed of errors, and some sections that are not necessary. So introns are the sections that are not necessary, and the exons are the sequences that are retained to create your final transcript. So you remove the introns and retain the exons. So that's one way to remember it carefully is that exons are called exons because they exit the nucleus. Because remember, the DNA is inside the nucleus, but the ribosome is outside. So there must be a way for basically, the information, actually the main role of the mRNA is to deliver the information from the gene that is inside the nucleus to the outside, to the ribosomes outside the nucleus. So that's why it's a messenger. So you have exons because those are the segments of the messenger RNA that is exiting the nucleus. Oh, by the way, when we talk about replication here, this growing strand that is being synthesized or transcribed, this is your mRNA. This is the messenger RNA. So anyway, the exons are part of the messenger RNA that is that exits, or rather that is retained in the transcript and exits the nucleus. Now, we have alternative splicing, which allows one gene to encode multiple proteins. So that means we have different sections that are being moved. So for example, we have one gene, so it has a combination of introns and exons. And then we have, from that single gene, you can create several transcripts by just changing what sections are edited out or what sections are being retained. So we call that the alternative splicing. So other exons are spliced together in various combination, and that gives various protein products on your gene. So variability. From a single gene, you have various protein products. So after splicing then, so aside from splicing, you have your capping and adding tails. Because again, RNA is less stable than DNA, and when it goes outside the cell, we have harsh chemicals and enzymes outside the nucleus, not outside the cell, outside the nucleus, that can degrade your RNA. That's why it must have caps and tails in order for the nucleotide not to be destroyed easily. So we have a cap at the 5' end and a poly A tail at the 3' end. When we say a poly A tail, it's just as a very, very long series of adenines added at the end, at the 3' end. So here is your alternative splicing. So as you can see, this is your DNA. So this is the promoter sequence, and this is your gene. So your gene contains regions called exons and introns. And these are copied in the newly transcribed messenger RNA. And then you have splicing. So you splice it, you remove the introns and retain the exons. And depending on the splicing event, so you have possibilities. Alternative splicing, although it's not always like that, but there are alternative splicing events. So you see, one messenger RNA can have 3 exons, or another have only just 2 of the exons. So it depends on the processing. Basically, how do you know that it will be this one or this one? This is basically part of the research called epigenetics, and it's still a growing research. It's still researching about this event, how and why they do that, or rather how and why the DNA or the genes do that, basically. And then you have, after this alternative splicing, you have your capping and adding of a tail. So you add a cap and a tail, and that is your mature messenger RNA. So in the next part of this lecture, we'll discuss about the genetic code. So this is now the translation step. So this information, how does it become protein?